with our 11.1 antibody production and vaccination. Some of the stuff we've talked about in 6.3, but it was really part of this AHL section. So a little bit of review, and we've talked about briefly about antigens and antibodies, particularly the different blood groups. So here what we see first is each blood group and what type of antibodies as well as antigens they present. So in group A, what they produce in terms of antibodies, as seen here, is they produce antibody B while they present antigen A. This is super important because we need to know our blood type before we actually have any transfusions, because if an A blood type individual was given a B blood type, what would happen is that their red blood cell for the B individuals, they present the antigen Bs. And then we'll just write B in there to make sure we know. And an A blood individual will be producing these antibody Bs. These antibodies will be able to attach to the antigens on the B blood. So this would be bad for the individual. So it's really important for us to recognize which types of antigens and antibodies everybody has, which is why it's important as well for AB group, so group AB, because they don't produce any antibodies, they are considered the universal recipient. They can accept both A, B, as well as O blood. And this is because they do not produce any antibodies as seen here, and possess blood that has antigens A and B. In the reverse fashion, we see O blood type group, because their blood type, they don't usually present any antigens. So they produce antibodies for both A and B. They're considered the universal donor because they can give blood to both A, B, and the special combination of AB blood. And they cannot accept A, B, or O blood. And lastly, we can just go over another example. So, for example, B blood individuals, they can accept O blood because O blood has no antibodies, antigens, and they can also accept other individuals' B blood. And this is again because other B blood will also have antigens of B, but no A antigens. Moving on to the next thing is a production of monoclonal antibodies. Monoclonal antibodies, again, antibodies are proteins. They have this Y looking like structure, and antibodies are used to detect antigens. So monoclonal antibodies often is written as little m and then A, B for antibodies, and then S for plural. Mono meaning single, and then clonal meaning clones. So you do need to know this process. On the next slide, there's going to be a written summary. But what's really important for us to talk through is that we often use mice for this. We're going to be inserting an antigen into our mouse, and we're vaccinating them and causing immunization or immunizing them. This immunization and the function of presenting the antigen to them is going to cause a immune response for the mice. Because they're going to produce an immune response, we're going to be able to see the cells be produced, and we're going to have antibody forming cells. So this is why here now we have these cells that have produced these antibodies. Because antibodies forming cells eventually will either die, because they'll die off after being used a certain number of times, or they're going to stop responding because the mice has recovered from its sickness to the antigen. We need to be able to produce a unlimited source of antibodies. Therefore, we use tumor cells. Tumor cells are often known as melanoma cells. Melanoma. So melanoma cells are used because they are effectively cancer cells or tumor cells. We use these melanoma cells, and we're going to also use the antibody forming cells, and we're going to fuse them. Just to note, the antibody forming cells are actually taken from the mouse's spleen. So they take the mouse's spleen and those antibody forming cells, as well as the cancer cells, and they combine it to form hybridomas. The hybridomas are then screened for the productions of the desired antibody. This is important because all the antibody forming cells can produce a number of antibodies, but sometimes we're only very interested in only one of them. This is why later on, once we've found the antibody that we're looking for, we're going to select that singular hybridoma, and because it's been fused with this tumor cell, it's going to produce these as many as we would like. So we're going to undergo clonal expansion. Eventually, we will just have the monoclonal antibodies. So it's great technology that we're able to use it. And some cons of this technology is that it's very costly and very expensive. It's also very time consuming. It could take multiple weeks, if not longer, to actually go through this entire process. And then lastly, it also consumes many liters of culture. So the culture is the media that they use to grow the hybridomas. So here's a little written summary that you can review on your own time. So what is the application of these monoclonal antibodies? Well, one of them is for rabies vaccination. So we see these. So the antibodies are actually located inside the vaccination or the immunization that is going to be administered. So dependent on the country, here in Canada, we actually provide individuals who work with animals or are regularly exposed to rabies or possibly exposed to rabies, we provide them a pre-exposure vaccination. This is just to prevent any effects even when exposed. But usually if you've been bitten by, if even suspecting exposure, individuals will go and seek additional vaccinations. So here are just two brands that are offered in North America, Rabivert as well as Immovax, for individuals who would like to be administered vaccinations against rabies. The second application that is also very important and likely would be used by anyone who is thinking of family planning. So one of those things that happens is that during pregnancy, there's a hormone called HCG produced. So this hormone is actually only produced, I don't know why my pen's not working, produced by the corpus luteum. We're going to learn more about this, but this is a structure located inside the ovary. So if this was the ovary, and during the follicular development, and then the follicle gets released down, the oocyte gets released down the fallopian tube, there's a structure that remains there called the corpus luteum. That corpus luteum is also going to be involved in the production of HCG, particularly during implantation. We're going to talk more about that later on. So here in this graph, what we see with HCG concentration in weeks of pregnancy, we see that it's constantly rising already shortly after fertilization. So tests can be used around 9 days after fertilization. If you take it any earlier, you may not have sufficient HCG concentration to be seeing any results. What's really important is that it peaks at 12 weeks and it decreases, but it's still going to be high enough that the pregnancy kit will be effective. Here we have a pregnancy test. And the idea of peeing on a stick comes from here because it is a stick structure and it's going to wick the pee. And this is going to be used by any individual who believes that they're pregnant. And what they'll do is first pee on this section, which would be exposed in this area. And the pee is going to wick through until this section. There are antibodies, so the HCG is going to bind to the antibody. So the antibodies are what is placed inside this pregnancy test. So we've produced these in the lab and then we've placed them onto the stick. So as the pee is going through, it's going to possibly associate with the antibodies if HCG is present. And then we're going to continue wicking across the pregnancy test. And then here we encountered an immobilized antibody test zone. So here, if there is an antibody that is activated by the HCG, it is going to bind and then cause the test line, the test line here, to become apparent. There's also a control line. This control line on this side is super important because this control line ensures that the monoclonal antibodies work. So if they work, they're still going to bind and then make sure that line is there. This is how we know when we read the pregnancy test whether we're pregnant or not. So pregnant is a double line, whereas not pregnant is a single line. And again, that's to test to make sure that the P does wick across the entire pregnancy test. The last thing we need to talk about today is, again, something that we've talked about, but just making sure that we're going to really solidify our understanding. So our very important system that is part of our immune system is a lymphatic system. The lymphatic system is what is drawn into the yellow, and what some of you might already know is our nice little tonsils that often get inflamed, and some of you might suffer from a lot of tonsillitis and have them removed, and they're actually part of the lymphatic system. The spleen, which is why the mouse's spleen is used, because it's also part of the lymphatic system where a lot of those antibodies are produced. We also have the thymus, lymph nodes, which are actually spread all throughout your body as indicated by all the little dark dots here. And lastly, we have lymph vessels. What you'll also notice is that the lymphatic system actually follows the circulatory system, which we'll talk about at a later point in time. What's also really important is that the bone marrow within all our bones produces B cells and T cells. So again, our big category is called lymphocytes, and there's many different subtypes, but we're going to talk more about B cells and T cells. So if we were to categorize lymphocytes goes into a branch to form B cells. The other branch is called T cells. And under B cells we have plasma B cells. And then the other one is memory B cell. T cells, we have two types that we need to know for IB. First one is helper T cells. And the other one is called killer T cells, sometimes known as cytotoxic T cells. And just to make sure that we do know everything for that, plasma B cells, they are large cells with large endoplasmic reticulums. The purpose of this endoplasmic reticulum is to synthesize proteins. But more importantly, synthesize antibodies. So those antibodies that are going to be synthesized are going to be specific to exposed antigen. Additionally, those plasma B cells, they respond to helper T cells. So the helper T cells help activate these plasma B cells further to produce more of the antibodies and release the antibodies. So here's a plasma B cell producing lots of the antibodies. The next one, under B cells, is the memory B cells. The memory B cells help with long-term immunity, and as discussed previously, it can provide 10 plus years of immunity. Just depends on the antibody. They also remain in the bloodstream. The next one is helper T cells. Helper T cells, sometimes if you watch videos online, they might be referred to CD4 plus cells. They're the exact same thing. They're used to determine the immune system, whether it's healthy or not. Used to determine what that means is that if ever you went to the hospital and they were looking for signs of infection, they would actually test your blood cells. And what they're looking for is around 500 to 1,000, 5,000 CD4 cells per millimeter cube of your blood. How are they helpful in your immune response? They become activated when macrophages, and again, remember that macrophages are part of digesting the proteins the pathogens. And after they're exposed to the pathogens, they will express them on their membrane protein, plasma membrane surface. The helper T cells detect this when bound. And they get bounded by these MHC complexes, which I think is beyond this course, but they are recognized, is all you need to know. This is then helpful because helper T cells go back and also activate more plasma B cells to produce more antibodies. Activate Lastly, we have killer T cells. In videos, you might hear them referred to as cytotoxic T cells. Some videos will refer to them to CD8 plus T cells. All the same types of cells. Purpose of these cells predominantly is to recognize and bind to antigen presenting pathogens, and they will release perforin, which is going to perforate their cells, and then also release cytotoxins. toxins. And that is going to cause cell lysis in the pathogen. So now that we've talked about the 4 different types of major cells, make sure that you understand where they fit and how lymphocytes are all divided. And just also remember that macrophages are also involved, and they're a type of white blood cell. Lastly, we just need to talk about antibodies and their purpose. So what's great about BioNinja, if you haven't used it, we have a nice mnemonic. We have panic is what is caused by the presence of antibodies. First thing is precipitation, meaning that soluble antigens make soluble antigens insoluble. So precipitate or create a solid, as many of you chem students will know. And that's really important because then we're going to be able to detect them. Agglutination, this is something that we've seen before. This is going to cause clumping or sticking together. And when the body finds that all the pathogens are stuck together, it makes it much easier to attack them and neutralize them. Neutralization is the same one. Masks dangerous parts of the pathogen. So possibly some pathogens are releasing. So ensures safety. Inflammation. We're going to talk more about histamines later on, but inflammation, it triggers histamine release, increasing immune mobility. So we've talked about inflammation and how if we inflame the area and swell, it's going to allow for more diffusion and osmosis of the white blood cells and the macrophages, as well as any of the lymphocytes that we need. Complement is complement protein perforates the cell membrane. So this is predominantly the cytotoxic T cell at work. And it's going to help cause the perforation as well as cell lysis. So panic, P for precipitation, A for agglutination, N for neutralization, I for inflammation, and C for complement. That's it. Have a great day.